How's everyone doing today? Okay, so I see a few of you are here, but all right, great to see you, Durba. How's everyone else? Okay, Asish, yes, if you cannot join the live session, you can watch the recorded session. I will show you today how to do that. Dr. Cheney, hello. All right, hello everyone, and it is 3, so let's get started. We have a lot to cover today, so hopefully this is going to be a useful session. Today, we're going to do a few things. I'll start slow. We'll get to a practical hands-on session. Before we begin, I want to show you where you can always find the information that you need if you are not present here in person. So remember that all of the program information is going to be found on the program page. You can search here for the program page, LBRN OmicsLogic Data Science. And if you are a member, so let me just copy this over here. You should be able to see not only what I see in the beginning, but also you can see that the first session is already posted right here. So Asish, yes, this is where you can find all the previous recordings. And of course, today's session link is also given right here. So hopefully that answers that question. Are there any other questions before we begin? All right. So if there are no other questions, let's start with the session today. So first of all, the OmicsLogic Data Science program is designed to give you an overview of different types of omics data, which is what we will do today. And in general, it is separated into 2 different parts. The first part is to kind of get you started with the data, knowing how to handle data, knowing how to explore data, visualize data. So that's what we're going to be starting off. And then in the second part is going to be about machine learning, statistical analysis, and discussion of how these methods could be used in a variety of different projects. So today's session, the goal is to really discuss again the, oops, going too fast. So today the goal is to really look at some of the data types and the project examples that we're going to be using in this session. And one of the things that we started talking about last time is that it is very useful to get started for those of you that don't have a lot of biology background by going through the Bytes and Molecules course. If you're more coming from a engineering perhaps background, that would be the place to start. In the Introduction to Bioinformatics course, there's a survey which you can fill out to let us know about you. And we will be looking forward to connecting with you and learning more about your goals and objectives. And so hopefully you can fill out that survey. And then the material for this course conceptually that you can start going through is found in this course 7, BioML Machine Learning for Biomedical Data. So maybe I could pause here a second and ask how many of you actually had the chance to go through any of these courses and try to complete some of them. And maybe if you have done at least one, put number 1. If you've done more than 1, then maybe put 2. If you've done nothing, then put 0. That's also okay. All right, excellent. All right, thank you. All right, so for those of you that have not started yet, I recommend trying to do that maybe over the weekend. All right, thank you, Ethan, Hadaya, Kisembo. Okay, so if you haven't really started, please try to get started with those courses. They will supplement a lot of the things that we do here in these sessions, and we'll give you a sense of where we're going. Now, the other part that I wanted to mention is that we will rely on the on the T-BIOINFO platform to process some of the bigger data sets. Has anyone, and we typically see that a lot of people have trouble logging in, or they don't really understand what information to use for that. Did anyone have trouble logging into the T-BIOINFO platform? And if yes, is it that you can't get in, or once you got in, you didn't know what to do? So maybe you could also share that just to make sure before we go on. We will try looking at some of these methods today to kind of talk about how they are useful in what we're going to be doing. Did anyone have any trouble, or did anyone try? Okay, so Rebecca, maybe you could try that now. This is the link. Okay, you can try logging in with the same credentials that you have on the Learn OmicsLogic portal. This platform allows you to process large data sets. Each one of you has an educational account, which means that I will be going through some examples and showing you how the data goes from raw to structured in today's session and also next session. So please try and let us know if you have any issues. Finally, there are some exercises that allow you to play around with coding on the portal. And we see that a lot of people get confused between Python and R, and maybe something didn't work exactly as expected. The reason why this is useful to have on the portal is because we want to be able to do some basic steps like loading data, visualization of data, and working with different types of data to explore and understand what it means and know that we are doing the right thing. So they are designed to get some similar outputs that we are expecting so that we can check that your code works. Yes, you should have an account that is the same account like on the Learn portal. So if you don't have that account, you can't use the same password and it doesn't log you in, then you've got a problem. And so if you can share your email address, I'll make sure that our community manager gets back with you to set that up. Okay, so do let me know if you run into some challenges. So finally, after you are done with the basics, you can go through some of the project examples. And these project examples illustrate some of the methods that could be used in application to a variety of data types. And importantly, not only can you do projects that others have done, but you can also try to develop your own project. So at the end of today's session, if we have time, I will show you how to do that. All right, thank you, Hidayah. I will ask our community manager to look into that. So what was the assignment the last time? The last time the assignment was number 1, to complete your profile on learnomiclogic.com and complete Introduction to Bioinformatics and or Bytes and Molecules. Number 2, explore server T-BIOINFO by playing around with demo pipelines. And number 3, install R in RStudio and or Python and VS Code on your computer. So how many of you have done, we already talked about 1 and 2, how many of you have done number 3? So how many of you have R and RStudio on your computer? So if you have R and RStudio, thank you, put number 1. Okay. Okay, Kisembo, if you are having trouble logging in, then please put your email address. I'll ask our community manager to assist you with that. All right, excellent. So, all right, so if you have R in RStudio, this is what we're going to be doing today. In the next session, we're going to be playing around with Python. So please install Python and Python 3 and install VS Code, Visual Studio Code. All right, and thank you, Asish and Kisembo, for your emails. We will check that as well. Is everyone here familiar with the interface for R and RStudio? So if you're already familiar, then let me know. Maybe also put number 1 or something. No? Okay, so so let's go through just to kind of review. So let me, if you have R or RStudio, please open it. So I mean, open RStudio. And let me know once you have it open. Okay, let me know once you have it open, maybe put in open. Okay, I'll give it just a little bit of time for all of you to have it in front of you. So thank you, Hidayah and Rebecca. Anyone else? Okay, for those of you that are not familiar with the interface, basically you have here 4 different sections. And these sections, this one is where you write code. So if you don't have anything open here, you go to File, and you can put New File and R script. So that's what we're going to be doing. We'll do this in a little bit. So here I have just an open file. It's really a text file with an extension .r. So here you're going to be writing the script. So the recipe for the computer to execute. Here you have your environment. In this environment, you will have different objects, tables, etc., that you have loaded into your project. And so here you also will find history, connections, and tutorial, but we will just deal with the environment right now. You can also import the dataset directly through here, and you can clean your environment from all the previous things that you might have had from a previous session. Here down at the bottom, you will see files, which is just a connection for your computer. So you can see that you are in your computer and you can set a working folder, which is important because once you have your working folder set up, it's much easier to call the files that you will need. Right here, you have set as a working directory or go to working directory. And so depending on where you are, this is where you want to deal with the different files that you're going to be using in your script. As you draw some plots and outputs, you will see them appear right here unless you specify to save them. And here you will see the different packages that are loaded. So you can install or load packages by running that here in the script or right here in the console. And if you have any questions about what package can do and how to write a specific function, very useful to write it in here. Some of the non-visual plots, like tables and things like that, could also be opened right here in the viewer. Okay, so hopefully this is like a very short, brief overview that helps you understand the environment. But as we work through the different exercises, you will definitely see how all of this connects together in an environment that is easy to use. So how many of you remember what we spoke about last time? Does anyone remember what was it that we talked about last time? What was the main kind of message from our last time? Import data. Perfect. Yeah, we did that at the end. What else did we talk about? Okay, one of the things that we talked about in our last time is we talked about what data science is and how is it going to be applied to what we will be doing in this program. And one of the things that we talked about is how we go from data to insights through specific steps. And one of the important steps is that we can make an action or we can understand a data set once that data is analyzed and structured in such a way that we can deal with it. And so before we get there, we have to input and load data, right? So importing data, whatever data we have, and that data could be tables already, but most likely it is not really structured yet. And our goal is to analyze this data to identify some relationships, some meaningful relationships. And so before we can do that, we have to deal with unstructured data. So what kinds of unstructured data are there? For example, media posts, emails, online reviews, videos, images, speech, and sound. All of these are unstructured data. We'll take a look at that in a second. There's also what's called semi-structured data. So the semi-structured or semi-unstructured data means that somebody already did some organization to it, but it's not really ready for direct analysis. And then structured data typically would come in some kind of a clear table, essentially, right? So to get a sense of how to do this, and Durba, I will get to your question in just a second, let's do a little exercise. So here I have an example of unstructured data. The unstructured data here comes from a chat, right? So you can see right here, I have a time point, who it's coming from, and their reply. And what I would like to do to analyze this data is to structure this data and understand what are the proportions of participants that answer a specific question that I have, right? So I really want to get an answer from all of these different variations. I want to get a specific answer. So how do we go from that? And today we'll do a little exercise in R to get familiar with how all of this works. So the first thing that you will need for that is a file. And I'm going to give you the link to that file. Okay, so what you do is you take that link and you save it. Save it where you know how to find it. Okay, so let me know once you have downloaded the file. And right here in this box for your files, you have to find the folder that you saved it in. Okay, so you can look, where did I save it? Oops. Okay, and once you get there, let me know. Yes. Okay, how many of you got the file? Okay, so once you get that file and you found the folder, you can see I had it kind of buried here in all kinds of different folders. Now you want to click right here. Yes, just save it as it is. Don't change anything. So right here, you have this gear icon, and you go to more, and you say set as working directory. All right, so let me know. Yes, you save it as it is. So let me know if you got there and you have this file. So how many of you have it ready? All right, thank you, Hidayah. Okay, so Rebecca, you go to the home, right? You go to your main folder and you have to find the downloads in this path. So let me set it as working directory. Okay, so if you go all the way to your home, right here, it should say home. You should find downloads right here, downloads. And then once you're in downloads, just set this as a working directory. Okay, could I ask the others? It doesn't show up. Are you in home? I mean, you must have, maybe save it instead of, okay, thank you, Amrit. If you maybe save it in some other place, maybe put it in on the desktop or somewhere else. What about Durba, Sish? Do you guys have it? Ethan, Dr. Cheney? Okay, thank you, Ethan. Sish, yep. Okay. Ayomide. Adetayo, yes, you're here. Amrit, Hidayah, okay, great. Okay, save it as a working directory right here. You go to this gear and you say set as working directory. And I'm going to go to my working directory, so I have it right here. Let's take a look at what this file actually looks like. So you can see that this file is semi-structured. First of all, we separated already the timing of the answer. We have the student number, and then we have some kind of a score. So these were some questions that we asked and they replied, but as you can see, not everybody followed directions. So we have lowercase c, large c. We have some people that put in the full, right? Just like we're doing now. If I ask you to put 1, some people say yes, right? If I ask you something else, somebody else puts something else. So there's no control over this. That's how the data comes out. So now I need to structure it, right? And structuring means coming up with some kind of a taxonomy, essentially. So for example, here, I made a formula. And the formula basically said that if a person says undergraduate, so here I have an answer key. If it's undergraduate degree, it's 2. If it's high school, it's 1. Graduate degree, 3. Postgraduate, 4. And using this formula, I can put all the numbers and save all of the information right here in sheet number 5. So what does this data represent? This data represents the number of students that I have and what is their level of education. Now let's try to load this data and try to find what we want to understand about these students. So what do we do first? What kind of a data is this? What format is this? Who remembers what format, and how do you actually open this format? And by the way, we don't have to just chat. You're free to unmute yourself. Okay, it is organized, yes. But what format is it? What format of file is it? Exactly, excellent. So to open that, we need a library. We don't just use the base functionality of R, we need a library. And this library is called OpenXLSX. Okay, so if you write open or library OpenXLSX, to run this library, you can go right here and run it. Okay, so that will execute this code. And sometimes you will see that you don't have this library. Okay, so what to do if you don't have a library? You go to Tools, and right here you can install a package. And you find it in the list of packages, and you already have a path to where all your data is stored, all of your packages, and you just click install. And so down here, you can see everything that you tell the computer to do, it does. And so here, when it's running, you probably saw there's a little stop sign, and that means it executed this, and you can see that it installed it. Okay, so now if you run it again, you see that it loaded the library and there are no errors. Does everyone have the library ready? So maybe, okay, excellent, thank you. Rebecca, are you getting an error? Rebecca, did you get some kind of an error?
I don't even know where I'm looking. What am I looking for? So I pasted library, open XLXS, whatever, right? So now what do I do?
You run it like this. You click on the run.
Where?
Button,
Run
right,
where?
right
Oh,
here.
okay. It's like
Okay.
tiny. My screen is tiny. Okay, I run it.
What did
And then
it
how do
say?
I know?
What does it say down here?
Nothing.
So it just says like this.
No, it doesn't say that.
Are you sure that you're on console?
Yes, it says console. Yes, I'm on that tab. If I have to install something, what does that mean?
Okay, so what you can do is you can copy this, just the name of the library, go to Tools, Install Packages, and then right here it has packages, you install
Too
this.
fast.
Here, Tools, Install Packages. And Hidayah, do you want to install from sources the package which needs compilation? I guess yes. Okay, so once you find it in the packages, you just click on install. And by the way, I don't know why you would not have your console, but let me know if that works for you, Rebecca. Okay. So Hidayah, did you get it installed? And how do you check that it's installed? You just run it. No? What happened when you got that message? Did you say yes? Okay, for Durba it worked. So Hidayah, maybe you can just unmute yourself and quickly explain what happened. So nothing should happen, right? We didn't do anything. We just loaded a library. Okay, so nothing really should work, but we'll find out in a second if it's working or not. So the second thing that we want to do is we want to create an object. What is an object? Anything can be an object. I can make an object, whatever name I want to give it, I can. So I will call this object education level. Now in R, your capital letters are important. So if you make this education level with capitals, then you have to refer to it by capitals as well. In R, we use this to mean equal, but really you can also use the equal sign. A lot of times people use this just because it's easier to separate when a variable is equal to something as opposed to loading something into an object. So right now we created an object and we want to load into this object the answers that we have, this read answers to question. So the function, so why is it called a library? Because it contains a library like a dictionary of different terms or functions. And the function that we will use is read.xlsx. So all it does is it reads this type of a file. And then we have to put in here the name. So answers to questions.xlsx. And we want to specify which sheet. So which sheet did we actually use? The level of education, right? Education level is sheet number 1, 2, 3, 4, 5. So sheet number 5. So right here, you put in sheet equals to 5. Now, to try that it works, always when you want to try something that works, you can select it and you can click on run. So what happened? Now in our working environment, we have here the education level is an object. And you can see if you hover over it that it says data frame. Okay, so how many of you have the data frame working? I can paste this for you. All right, Amri, thank you. Thank you, Ethan. All right, Madeline, Asish, Rebecca, did it work for you? Okay, so again, the working directory, once you find the directory, you just click right here and you say set as working directory, but you have to find the directory here first. You have to see the file. Okay, I see that maybe. Okay, then find the desktop. So go to your desktop and then set it as working directory. Okay, so for those of you that are still struggling with some of these technical things, we will have a recording of this. Maybe you can try that again, or we have some exercises for you to try. You can try and do this on the portal. That way you can get started. All right, what do we want to do next? The next thing is that we want to load a particular type or particular column from here, right? So what do we actually need? We need this column, score. How do we load a score? We can say, for example, make a table or make another object, or let's call this score. And load in here education level, and we use the $sign. And you see when I put the $sign, it already tells me what are the different columns that I have. So time, name, and score. Okay, so yes, in the beginning, when you install R, it does automatically set the directory for your working directory, but you can always find it right here on top and change it. All right, so here we want to take the score. So now if we run this, you can see that now we got the numbers. We had in total 26 students, and now we have the answers for those students right here. Okay, did everyone get the score? All right, now, okay, yep. Okay. All right, so we have 1, 2, 3, 4. Anyone else? Okay, is everyone else still stuck, or do we have, so Rebecca, did it start? Okay, Rebecca, it works for you, great. Madeline, did it work for you? Okay. Kisembo, maybe some of you already know this. So Dr. Cheney, does it work for you? All right. So not yet. Okay, so let me know where exactly you're still. Okay, waiting to complete install. Okay. All right, so by next time, hopefully this is already working for you guys. I'll show you in a second how we can perform all the same in the OmicsLogic portal, so you don't have to figure out all of the installations. So now let's create a plot. Okay, we saw how here we have our environment. Let's first actually just print it. So you can say print, and for example, the sum of all of our scores, score 1. Okay, now oftentimes when you get some kind of a error, it is because you did something wrong in writing. Maybe if I put here lower capital S and I run it, you can see error in print sum score, object score not found, right? I don't have an object. I can see here what kind of objects I have. It's not there. It doesn't know how to do it. But if I do this correctly, okay, and I print it, you see that the total is 79. Now it's meaningless. So this is not really useful. So I can just delete this. But let's try to create a plot. Okay, what kind of a plot would be useful to visually see this data? What do you think? What kind of a plot do you think would be useful here? What would be a useful way to look at this data? What do you guys think? Bar plot. Okay. So what would you like, Adetayo, for this bar plot to show? What would be a meaningful way to show this data? Should I just have, okay, Kisembo is saying histogram. So I could look at the distribution of these numbers. I don't know how useful that is. Maybe it will give me some sense which numbers I have more or less, but it doesn't seem like I have continuous data. This is really categorical data. I only have from 1 to 4. And if I want to look at this data in a meaningful way, what I would do is I would actually, yes, the number of students that had each score, right? So I would want to look at how many are 2s, how many are 1s, how many are 4s, etc. right? So instead of just a bar plot, I'm actually looking to summarize the data in some meaningful way. And so to do that, first of all, we need to put our put this information into a table. Okay, so for example, we can say table 1, and then put here table, and we can put education level score. So if I do this, you can see that it is a table now. So this object is called a table. So it's very similar thing to what we did here, but it's really a table. And once I have that table, I can just say table 1. And if I run it, what will happen? You can see that now it is organized into how many I have of which. So I have 2 of 1s, 6 of 2s, 7 of 3s, and 11 of 4s. Okay, so it's kind of exactly what we need, right? Okay, did everybody get this table 1? If you want to try this and you don't exactly want to look at my screen, here's the latest. Okay, did everybody get this? All right, thank you, Amrit. Ethan? Adetayo? Thank you, Ayumide. Anyone else? Madeline, Asish. All right. Who else do we have? Durba. Okay. And if you're still waiting on this to install, then unfortunately we'll have to wait until next time. Now let's try to create a prop table. So what we can do is we can, first of all, say prop table 1. So what did this give us? It not only counted how many do we have, but it calculated the average, the percentages for each category, right? So the categories are 1, 2, 3, 4. This one is 8% almost, 23%, 27%, 42%, right? So it's useful for us to know how to do this. And now let's create a bar plot. So if we do a bar plot of table 1 and let's run it. So we get a chart, or we have a plot, right? Right here we have this plot tab, and it shows us in a visual format what the data looks like. So just by looking at this, what number has the most? Number 4. What number has the least? Number 1, right? Did everybody get this?
Yep.
All right, perfect. Error. Okay, Rebecca, what does the error say?
Error in table education score object education not found.
Okay, do you have education level? Is it exactly like this? Because it's important to make sure that all of your spelling here is correct.
Huh, I don't know. I thought I copied and pasted all of your stuff.
Yeah, but it could be that I just gave you a portion of what I had. I didn't give you the whole thing. But if you want the whole thing, I can give you the whole thing. So copy and paste this, because right here, you have all of your objects. So if one of them is named differently, it's not going to work.
Okay.
Let me know if that works.
Do I have to delete everything that's in the box before I copy and paste what you just gave me?
Yeah, this box should just be what I gave you.
Okay, we'll start there. Sorry.
All right, so the culmination of this
Still have an error.
is still an error. Well, what is the error
Yeah.
say? What is the error say?
Error in table education score.
Education level score. This is not finding this. I think.
guess.
What do you have right here? Do you have education level like this?
As far as I can tell, it looks like that. Yeah.
And do you have this score right here? Does it show score?
Yep. And then I have values, score, numbers 1 through 126, 2, 2, 1, 4, blah, blah, blah. And then table won't do it.
Can
I don't
you
know why.
can you copy exactly what is the error right here? You can just copy and paste what it says.
Sure.
Object education is not found. Okay, and so it's because it should say education level, not just education, but education level. See, somehow.
Yeah, it does. It does.
Well, but it tells you that object education not found because your object is called education level. So you have to, are you running it in pieces or something?
I don't know what you mean by running it in pieces, but I don't think so. I copied and pasted what you put in the chat and I put it in. And as best I can tell, it looks like what's in your window. And now that's what it's telling me. Am I supposed to install something to do the table? Maybe I need to install something.
No, you don't need to install anything.
Okay, I have no idea.
Well, it just tells you right there that it shows us what's in your script, right? And if you just read it, it says that your line says table 1 is table, and then in parentheses, education, $score. And so you don't have an object called education. That's what it's complaining about. So why don't you just go in there, or you can also copy and paste what you have in here, or maybe you can share your screen if you want to, and we can see what the error is.
Can I share my screen? I mean,
I think you should be able to, yeah.
well, it says I can't share because you're sharing. But
I'll stop sharing.
okay.
Go ahead.
Share screen. Let's pick the right screen. There we go. I mean, is it sharing? No, share. Click share. There we go.
Okay, so now can you run it?
Click this run, right?
Yeah, but just when you click that, that's just the selection. So you got to select everything and then run it.
Oh, there's the missing piece. Okay, how about now?
Yep.
Okay, I'm so
There you
sorry. All right.
No problem. All right, so now we got it working. Now let's just summarize this by adding a little bit more information to this plot so it actually makes sense. So the first thing that we want to do is we want to give it some color. I like colors. So we can say col equals, col stands for color, rainbow. And the length, so the number of colors that we need is going to be answers. Sorry, we actually need to say how many, well, let's just do 4 because we know how many we have. So we'll just put 4. And then we'll give it a title. So when you say main will be the title. So what is the title of this plot? The title of this plot is summary of maybe student answers. Okay, so now we can also add what these answers are. Okay, but to do that, I'm sorry to make it a little bit confusing. Let's add the answers. So what are the answers? The answers, what are these numbers? 1, 2, 3, 4 are right here. 1, 2, 3, 4 have the answers right here. So how do we get that? What we need to do is we need to load not sheet 5, but sheet 4. So let's create another variable. And we will again do read XLSX. Again, let's write it out. Answers to questions. We can just copy this. But sheet equals 4. Okay. For those of you that want to try this later at home, I will share this in the chat. So just make sure you add it in the right place and let's run it. All right, so now I have answers 1 and it has 4 objects of 2 variables. Why 2 variables? I have answer and number, right? And then once I have this information, I can actually use this information to replace these 1, 2, 3, 4. Okay, so the way I would do that is I would add here names. So these would be the names of my things. And I will say answers 1. And the column would be answer, right? So let's try that. So this is what I have now. Let's put it right here. All right, run it. And let me know if you got the same. All right, great, Madeline. And read. Well, it looks much better, right? Okay, just give you a couple more minutes. If you're having an error, do let me know. Ethan, thank you. Adatayo, Asish, Rebecca. Okay, Ayomide. Yep, thank you. Durba, is it still okay? You probably. What about Fiinfolua? I'm not sure if I'm pronouncing that correctly. Dua, Hidayah, Kisembo, let me know. Okay, so if you want to do this later at home, what can you do with this? What you can do is you can just save the whole script. Make sure that you save it in a file. Now try to play around with this, maybe give it some different kind of data. And to talk about different kinds of data, which is what we will discuss today, and I will try to do this fairly brief, we want to talk about, this is just an example how we went from fairly complicated semi-structured data that looked like this in the beginning to something that we were able to summarize and then visually present to get an answer. The question was, how many or what is the biggest category of people that we have? And the answer is postgraduate students. Okay, so now let's talk about what other types of data are we going to be dealing with? Because really, this is small data, right? This data is small. This data is simple, it's intuitive. We know how to look at this data and make sense of this data. What we don't know is what is in the big data, right? And so what kinds of omics data have we talked about previously? Can anyone remember what are some examples of data types that we talked about before? Does anyone remember? Yeah, what kinds of data did we talk about last time? Omics data, that's key. But what kinds of omics data do you know? Genomics, okay, good example. What else? Transcriptomics, metabolomics. One more. Metagenomics. Okay, excellent. So here we have some overview of different types of data. Phenomics, we can talk about phenotypes. If it's high throughput, that it probably is automatically generated. So this could be, for example, cell morphology. Genomics really represents genomic variants. So this could mean that it's categorical data, data that is A, T, C, and G, but it is across the whole genome or the whole exome. So that's why it's called omics, a collection of that kind of data. Epigenomics, epigenomic data could be ChIP-seq data, whole genome bisulfite sequencing data, all kinds of data. It looks like this. It looks like peaks in specific areas that are from a pull down of a protein that binds to a particular location. So you know how to look at structure of chromatin, or maybe DNA methylation, or maybe even miRNA regulation with microRNA. And then transcriptomics. So transcriptomics represents the expression of genes and isoforms, and we can capture the variability at the level, quantifying the levels of expression, or looking at differences in specific types of arrangements of exons and introns, which is called alternative splicing. Now, with all of this data, the key step is processing. So what we did today in the little exercise is we actually converted data through multiple steps from one way that it looks to another way that it looks, right? And that is the processing. We didn't do anything with the data, right? Before we summarized it, we were just transforming the way it looks. And so in the case of omics data, that's a very important step, and you have to know how to do it. And in our next session, we're going to learn how to do that to actually process this data. Why does it need processing? Raw data looks like this. Here you can see reads. These are the individual portions of data that are 150 to 350 nucleotides long. Each one of these reads contains sequences. But together, those sequences need to be analyzed because they map on some location of the genome. And now we deal with this huge data that could be gigabytes or even terabytes in size. And what do we want to get out of this data? Again, we want to get some kind of answers. So how do we go about those answers? Let's first of all talk about genomic data. Genomic data represents sequences of tcGNA, and the answer we're looking for are specific mutations oftentimes. So here you see a VCF file. Does anyone know what a VCF file stands for? What is a VCF file? What does VC and F stand for? Okay, VCF stands for variant call format. So instead of looking at all of the positions, we want to only call the specific positions. So what you can see right here, and this position right here is 7,674,220. So that position contains a SNP. It contains a mutation. And so instead of looking at all of the data, I just look at data. This is chromosome, for some reason, this says chromosome 17. Yeah, so this is, okay, so this is chromosome 17. And you can see that right here in this position, we find these two mutations. So this is the outcome of processing, just selecting this kind of data. Now, a question is, if we have this kind of categorical data, remember it's only 4 possibilities, tcGNA, what would be a way to convert this data to numeric data? Because really what we can analyze with a lot of the methods that require statistics and machine learning, they would have to be continuous. So the answer to that is that we can actually assign each letter a score. And the score is typically a result of analysis, considering all of the input data that you have. So how many of the reads, depending on where it is, how many, and which conversion it is, you can actually calculate a semantic score. What do you get as a result? As a result, for each position, you have a semantic score, so a number. And based on this number, you can now filter using some kind of a threshold and get a result. And then the rest of that information is interpretation. So how does a particular mutation, a change, can actually affect some kind of a disease, a phenotype? In this case, you are looking at sickle cell, and a mutation that happens in patients with sickle cell disease that actually causes this disease. So from raw data, going through the steps of processing, then selecting some kind of information that we need, and then going to interpretation. Another type of data that we will be looking at is, okay, so yeah, that's the mutation, that's hemoglobin right here. So this information is described in the genomics course that you will find on learnomiclogic.com. And it is really divided up into several different sections. Introduction to Genomics, Genomics 1, that talks about somatic mutations, and then Genomics 2, where you look at other types of ways that you can analyze such data. An important consideration here, as you can see, is that to make this data intuitive, we have to have a way to interpret this information. So how did we go from finding mutations to then knowing what is their effect on the biological interaction? we use the central dogma of molecular biology to connect the two, to say that we know that nucleotides form tricodons, those form amino acids. A change in a nucleotide could result in a change in the protein structure. That's why we want to associate how the protein structure changes because of the mutation that we found in the data. So in the same way, we now can turn to alternative splicing and expression of genes, or transcriptomic data analysis. So when we talk about transcriptomic data, we are really looking at how we can measure levels of gene expression or alternative splicing of specific transcripts or isoforms, so that we can also understand what is the outcome and associate that with a particular phenotype, or again, a condition or a change. So this requires, again, multiple steps in the processing pipeline. This is called a pipeline. What does a pipeline essentially do? A pipeline loads the data, just like we did in our script. We loaded the data first. Then we pass it on to a library, a function in the library. And that function performs maybe some very complex calculations on all of our data and provides some output. Once it does that, it passes this output on to the next job. And so from job to job, they are organized here in a logical way. So you go from pre-processing or cleaning your raw data, then to mapping it onto the genome, then constructing isoforms and saving them into a GTF file, then mapping the amount of transcripts that fall in a certain segment and quantifying that to provide us with a structured output. What does that structured output looks like? Again, like a table. This table now has different elements, and those elements are samples. All of our columns have sample names, and genes. And so at the intersection of gene and sample, we again have an expression level. Does anyone have any questions about this? Okay, so we're talking about how do we go from raw sequencing data or raw unstructured data to structured data that has continuous numbers. So these numbers are continuous. All of the information about transcriptomic data analysis is found in the transcriptomics course. So in between now and the next few sessions, you're welcome to take a look at some of those and see what's in there. Now, finally, the third data type that I want to introduce today is what's called metagenomic data. So how many of you know what does the metagenomic data represent? Adetayo mentioned metagenomics. So what kind of information can we find in the metagenomic data? Okay, any suggestions? What do you think metagenomics means? Okay, metadata is data about data. That's good, Ethan. Okay. Microorganisms. Okay, thank you. So what is different between metagenomic data and, let's say, genomic, transcriptomic, metabolomic, and others is that metagenomic data describes a collection of organisms and not just a single one. And instead of analyzing an individual organism, we're really looking at a community. And that community, instead of giving us the differences between an individual and another individual, really shows us something like a proportion. And that proportion, together with what we know about relationships between microorganisms at the evolutionary level, provides us with a sense of community structure. And that community structure is defined by how we think, how distant and proportionate they are to each other. Because the idea is that a single sample of microorganisms represents a community that functions together in a very defined way, in a stable way. And so when we sample that environmental sample, we are looking at a composition of what's called the microbiome. To achieve that goal, we have to disregard the small differences between what we have in our data and focus on groups. And these groups we call operational taxonomic units, or OTUs. Operational, how many of you know what a taxonomy is? Okay, so Rebecca, what is a taxonomy?
What a taxonomic unit is, or I mean, it's just a way to divide the bacteria in my mind. Maybe that's not what you meant.
No, that's exactly what I mean. So it's a way to look at our data and organize them into groups, right?
So, right, so genera or species or something like that.
Exactly, yes. So the taxonomy is how do we fit this individual that we might not know which genome it's coming from into some kind of a group that we can define by the level of taxonomy, species, etc. So to do that, we again take raw reads. Those raw reads contain a lot of variability. And one of the challenges is to distinguish between variability that is technical or comes from errors and find accurate data. And there's a pipeline or a whole section of different types of methods called Data2 that you can see almost exclusively focuses on going from raw data to assigning that taxonomy to a group of units. And those units are collections of reads. And then after we have them, we can now analyze them for the relationship or the phylogeny, how they are related to each other. And then we can create all kinds of visualizations using this phyloseq package. So what do we want to get out of this information? Typically out of this information, we want to analyze variability, or we want to organize our data visually into these groups that make biological sense. So we're really combining information that comes from data analysis with our understanding of biology that could be the reason for what we observe in the data. And so these are some of the plots that we're going to be building also in this program. And all of the information about this is going to be available in the metagenomics coursework. But it's important to remember that there's a lot of overlap. As you can see, we also want to get a table. The table now has, instead of genes like we had in transcriptomic data, operational taxonomic units, we have samples and abundance. So this abundance is a proportionate abundance to what we have overall. Also, we need to combine this with annotation, and the annotation provides us with that taxonomic organization. So for each one of these OTUs, in one table, I have how many, or the proportion that I have in the whole sample, and then I can annotate them based on what is known from a database. Okay, any questions about this? All right, so no questions about this. The process overall for each one of these data sets is we first have to process the data. Then we have to explore the data for variability that we can understand. And then we need to define what is the hypothesis. So in many cases, what we will do today, or not today, but in this whole program, is we're not going to go generate a hypothesis and generate data. We're going to be taking other people's data and using that data to ask questions about the data. So first of all, we're going to generate a hypothesis, and then we're going to find the answer to that hypothesis or interpretation that we can make sense of in a biological way. So this whole process of data analysis will be based on data that we analyze and mine first, and then ask the questions about it later. Now, here's an example of one of the data sets that we will be looking at. The example comes from a publication called Modeling Precision Treatment of Breast Cancer. It is a publication that takes a look at 70 breast cancer cell lines, or sorry, 84 breast cancer cell lines, out of which some of them have been tested with drugs, so we know how they respond to different types of treatments, and they have different types of data, different types of omics data. So it's a multi-omic data set. And what we would like to know is how, based on the data that we have, can we use this to predict the best treatment for a patient, right? So if you imagine a patient that comes and we have all of this data about them, how can we predict what would be their response for what we have, for their response ultimately when they're treated with the drugs. And so you can see here in the abstract and the results that are reported in this paper, how they used molecular profiling, which is the data that we talked about today, to identify specific features like genes to predict therapeutic responses, and actually find optimal predictors, which might be a combination of multiple and not just one, that will predict that response to treatment. Now, as you read through the results, you will start seeing what methods did they actually use to do that. And you will find words like support vector machines, random forest, and these are machine learning algorithms that we will learn how to use to be able to do same types of predictions that we see they did in this paper. So ultimately, why did we select these specific projects, and what are we going to be trying to imitate during this program that is dedicated to biomedical data? A lot what happens in the clinic is dependent on precise data that has been analyzed from thousands of patients. And so a good example of that is prediction of survival. So in many cases, we know that groups of patients actually respond differently to treatment, more, better, or worse. And therefore, over time, if we collect data about patients over time, we can know that some are going to be more at high risk or have a lower risk, which is associated with the outcome. How many years do they have to live after they were treated? And so here you can see two panels that are based on gene expression data, and that can separate all of the patients at an early stage, right here at year 2 or 3, very effectively to know their outcome over time. Another way that this data could be used is to select the appropriate method of treatment from traditional treatments that are available. For example, here, if we're looking at surgical, radiotherapy, or chemotherapy, all of these therapies come with an additional risk that could lead the patient to develop a different type of cancer, for example. And so knowing which type of treatment to select oftentimes is related to risk stratification. And risk stratification in this case is going to be defined again using molecular data. Now, the other example is targeted therapy. And targeted therapy means that we can take information from the molecular data at what's going on inside the cell and use that information to understand what type of proteins, for example, could be targeted. So how can we make a treatment that is going to treat specifically that patient and not anybody else? And so here you can see that the keyword here is biomarker. A lot of time, the biomarker could also be the mechanism of action. And so a good example for mechanism of action is when we have Herceptin. Herceptin is a drug that binds to the HER2 protein to stop the growth factor receptor from signaling a signal to the nucleus to produce different types of genes involved in growth and uncontrolled replication. So what we will do in the program is we will replicate what we did today, but instead of using survey data that we understand, we will take omics data. And from that omics data, we will go to use R and Python. Today we did a little bit of R. In our next session, we will do a little bit of Python to load the data, visualize the data, and see what does this data actually mean. So I think at this point, that's all we had for today. I do want to introduce you to some of the data sets that we will be using. So if you can go with me to the server portal. Sorry. Okay, and I will show you where you can find some of the data sets that we're going to be relying on in this program. So please let me know once you're here. Okay, maybe put something in chat once you've reached the website and you're logged in. All right, thank you, Madeline, Dr. Cheney, Amrit, Ethan, Dua. All right, so when you are here, you will find that different types of data, NGS data, for example, transcriptomic, genomics, etc. You can find next to a particular section a dropdown. Right, so here you have different types of dropdowns. The dropdowns are for demo pipelines. The demo pipelines contain data sets that are already analyzed. Okay, so here you can see different project examples. These project examples, and you will do a lot more of this in the next session, but these project examples come with an input, all of the steps to process, and the output. So here you click on start. You can then go through the steps to pre-process the data. So again, these are steps in the pipeline. Then map this on the genome. Construct isoforms. Merge those isoforms into the genome transfer format, or GTF. Quantify the levels of expression, and you can see what that outcome would be is a table. And then when you click on end, right here in the downloads, you will find the information that you will need to process the data and analyze it. Okay, so how many of you got this result? Okay, just to show you what are the steps to do. Here are the steps. So let me know once you got this result. Does anyone need me to repeat this? Okay.
Yeah, I think kind of stuck on your previous screen.
Okay, so let's go to areas of analysis.
Yeah.
And right here under transcriptomics, you open this drop-down arrow.
Yep.
Open the RNA-seq cell line project, the first one.
Yep, it's asking for my credential. Let's see. It should be the same though, the other credential we use for Omics.
Yep.
Login, right? Yep.
All right, and then you start, which tells you a little bit of what the pipeline could look like. And there are multiple ones that you can do here. And that's how you can play around with this and get to understand the logical steps. For example, now I'll make a different pipeline. So from here, I can go to bow tie to T and just create a table of expression in a faster way. Click on end and get a result. All right, and then when you get the result, let me know. All right, excellent. Madeline got the result. So if you got the result, let us look right here, cell lines. Do you need the steps, Hidayah? Here are the steps. So start, Trim-O-Matic, PCR clean, bow tie to T, RSM expression table, end. Once you are done, download this file, cell lines, clean type txt. All right. And now let's try, just like we loaded the survey data, we're going to load in this information from this pipeline. Okay, so I need to know how many people got it. Madeline, Ethan, Hidayah, were you able to get this? No. Rebecca, where are you? Do you have the pipeline?
So I don't know what happened. So I went back to the top page. So I'm under transcriptomics and I click on what? Cell RNA-seq. What am I looking at?
Yeah.
That one, right? I click on that, right?
Mhm.
And then I get to this. And I'm supposed to click on what?
Start.
Start. A window pops up.
Mhm. Okay.
Okay.
Trim-o-matic.
Oh, do I just keep clicking? Okay. Okay.
PCR clean. Bow tie to T. RSM expression table. End. And you open this folder and download this file. Did you get
Yes.
it?
Okay. Thanks. Yeah.
All right, so once you have it, again, what do we need to do? Right here, we loaded some answers to questions. Let's create a new file, our script. Okay, and now again, we have to navigate to that folder, right? So since most of us saved it in the downloads, we have to find the downloads folder. And then here under more, say set as working directory. And now we need to open, sorry, let's create a cell types object. And you can just, go back here and kind of see what you did before. Now this is already a txt, right? So what we need to do is we need to say open. Open could be different type of open. So if you write this, you can see here different options. You can see all of these are options. It will tell you kind of what you are doing, or you can do load. So you can play around with this. And the file, remember, you put the quotation marks. Okay, so if we do this, okay, if you try to do this, remember that this will not work because of how we define what it is that we are opening. Now, how do you, and this will be your assignment for the next time, so I'm not going to give you all the answers in this case because you already know where to find the answers. How do you get this into your R Studio? Let's go to Learn, and we'll find a course where you can practice some of these things. So if you go here and you do data science, introduction to data science in R, okay, go to how to load data and check for variable data. Okay, and then here you will have some examples, and we're not going to go through them, including trying them yourself to actually get this data in and do the same thing that we did before, right? So your output should be just like we did here, but instead of answers to questions, we want to know how different types. So you will see here, there are different columns. You can see cell line name, you can see transcriptional subtype, cell line, cell type, etc. So you will have to figure out how from this data do I build a bar plot. Okay, so similar to what we had here, for 4 different types of cell lines. Okay, and these are normal-like, basal, luminal, and cloud and low. Okay, so your assignment is to find which type has the most, which type has the least. Okay, where do you find the answers on how to actually do this? In this course. And then when you want to visualize, you go through here and you follow this tutorial on how to visualize this data. Okay, does anyone have any questions? Okay, so if there are no questions, Kisembo, and I think someone else posted here, I have your emails noted down, that you did not have access, so I will make sure that you have access. Did everyone join the Slack channel? Everyone here, are you a part of the Slack channel for this program? Okay. Okay, so if you are not a part, then you need to go to the program page. Again, look here for the LBRN Data Science program. And once you're in here, you scroll all the way down here to the bottom and you will see the Slack channel. So if you click on Ask a Question, it will redirect you to the channel. Okay, which means that this will, if you have Slack installed, of course, then you should be right here, LBRN OmicsLogic Data Science Program. So please send me a message here. If you're in here, then just maybe reply to me or say hello or something. And then if you need to install Slack or something like that, let me know in the chat once you're in here. So I see that Amrit is here. Let's find Amrit. Okay, so there's Amrit. There's Madeline. Okay. All right, Ethan is installing Slack. Great. All right, perfect. So here, if you go through the tutorials and you have any questions or you're struggling with something, please let me know. My name is Ilya right here. So you can mention me and I'll be happy to help you with whatever you're struggling with. And another person that's going to be guiding the sessions, his name is Clinton. So you can mention Clinton here, right here, Clinton Kunha. He will be teaching the next session. Okay, so DP, if you gave me already your email address, it should be in here. I will reach out to you with the details shortly after the session. All right, so hopefully all of you are here. or soon will be. Okay, Christina. Again, Christina, the way to find the Slack channel is to go into the program page. So this is the program page. And once you're here, you go to the very bottom and it says, ask a question. You click on here and then it will open a redirect to this channel. All right, hello, Sri. Okay, and is Sri here in the session? Okay, all right, great. Got it. Got it, DP. Okay. All right, so let's see. We have here Amrit, Sri, or DP, Joseph. Let's see if everybody else can join us just to make sure. Okay, well, it's 4:30, so I think we're out of time, but I'm looking forward to interacting with you here. We'll have another session on Monday. So before Monday, if you could try and work on this assignment. So again, in here, in our studio, try to load this data and make a summary plot of the types of cell lines found in this data set. If you want to read a little bit more about this publication of where the data came from, this is the publication, just to understand what were they trying to do and what are the different omics data types that they used. I think a very fascinating, even though it's somewhat old, it's from 2013, but a very nice study that they did there. Thank you, everyone. Have a great weekend. I will try to address all of your questions okay, hello, Becky. So I will try to address all of your questions. If you struggle with something, then let me know here in Slack. Have a great weekend, everyone. Thank you.